Now it is. And it will be available afterwards via the Eastman YouTube webinar. And for those of you who are here, you'll see that link in highlights. And we'll also post it on the Grantham listserv. Our next speakers, I should say, for this is our January sessions. We're going to have how Eastman has handled the COVID-19 prevention and control efforts. And so Bill Madsen will be here, as will Steve, and Leslie and our health team. Hopefully, I think the three of them will be here, all of them here, to do a little presentation to you all and then to welcome questions. It's been quite a, I don't even know how many months now at this point, but in terms of trying to figure out how Eastman can do the best that it can to protect the residents that live here. And so it's been quite an effort, and I'm happy they're willing to share it with us. And I'm sure that some of you probably have some questions about how it was done, why it was done, etc. So that will be on January 8th, same time. And then on January 22nd, we're going to have COVID and the flu, you know, similarities and differences and why it's such an issue. And Dr. Al DiMaria, who is actually Paul and my former boss, but he's a former state epidemiologist from Massachusetts, is going to join us for that one. So more information from that will be sent out. And finally, I get to our speaker, who we're very happy to have with us today. And this is Dr. Iain Sim. He has a PhD in microbiology and 30+ years of experience in the pharmaceutical and biotech industry, discovering and developing new drugs in the fields of virology, oncology, and immunology. He came to the US in the mid-1980s, and he was immediately plunged, as I'm sure some of us old folks remember, into the first infectious disease epidemic, his first infectious disease epidemic with the emergence of HIV/AIDS. This led to his 15 minutes of fame with Tony Fauci, as we all know who he is now. And that was on a PBS produced program on AIDS. And yeah, we were all much younger then. And truth be told, Iain's was but a small cameo appearance while Fauci actually dominated the program. There followed a tumultuous series of interactions with members of ACT UP in New York, who those of us who worked in the infectious disease arena well remember ACT UP and Larry Kramer. He and his group demonstrated against the pharma company, which was Hoffman La Roche, where Iain worked at the time, and lobbied for immediate access to potential AIDS drugs that were in the development pipeline. And those efforts were an amazing example of how really getting out there and having a voice can make a difference. So his subsequent assignments took him to Tokyo and Palo Alto, California, back to New Jersey, and to Basel, Switzerland. In 2001, he moved to the Upper Valley, and he promptly joined a Massachusetts-based biotech company and became engaged in a DARPA-funded program on pathogen countermeasures, focusing on anthrax. He retired in 2012, same year I retired actually, and has been an avid supporter of OSHA ever since, our beloved OSHA. And on that note, I will welcome Iain Sim to do his talk on screening and testing for COVID. Thank you, Iain.
All right. Thank you very much indeed for that introduction. And it's a great pleasure to be here. I think I have the control and I have to pass my first test of whether I can successfully share the screen with my slides. This talk, as I think you've had others in the past, really sprung out of a course that we presented. Paul was one of the presenters at OSHA at Dartmouth. So if you like the content that we've delivered to you and I'm delivering to you today, then just a little pitch. Please consider the opportunities that OSHA offers in terms of education. We have a very full winter course catalog with over 1,000 people enrolled in the offered classes. That'll be starting soon. And then the spring class catalog is being assembled even as we speak. That promises to be another bumper crop of topics which cover the whole spectrum from I would say, from knitting to quantum mechanics. So if you can't find something in the catalog, we're not doing a good job, but I think you'll always find something here. So with that said, let me see if I can successfully share the screen, and we'll get on with the presentation. Let's see if we can get this up here. Okay, I hope that that is now visible to everybody. Paul, can you confirm?
Yes.
Good, okay. So we're going to be talking about tests and testing this afternoon. So let's get right into it. And so I don't know if it's the view that I have. Part of the screen is obscured here. Can you see it all okay? Paul, can you see the whole screen?
Yes, I can. Yes, I can see the whole slide.
Okay, then it's what I've got here. Okay, I'll press on. So why do we do testing? Well, our president weighed in. He felt that he was being embarrassed by the fact that we were doing testing and revealing cases, but that's the whole point of testing. Testing is intended to give us an understanding of the scope of the pandemic. And contrary to maybe what the president believed, that taking a test does not cause the condition, as this young woman in the bottom left here has indicated. Taking the pregnancy test doesn't make you pregnant. So with that as a sort of start off, our little bit of humor, let's get into this. So why do we test? So I've got a suggestion in the top right corner there of a young woman who's presenting maybe at the doctor's office. She's got a cough, maybe she's got a fever, some muscle aches, headache. Question is, what is the condition? And the reason why it's important to differentiate whether it's indeed the COVID-19 caused by SARS virus, or if she's just got influenza or maybe another respiratory disease, is because it does form the treatment. We do have treatments specifically for influenza virus, drugs like Tamiflu, which would not work if it was if your case was one of coronavirus. So it's important to know what the causative agent is, because the symptoms somewhat overlap to begin with. Of course, if it's coronavirus, then we want to isolate the patient and to consider the treatments which are available. And right now, in very short order, I mean, this is quite remarkable, even with the experience I've had, that in 9 months, we've now come to recognize at least 3 different treatment entities which could be used at different times in the course of the disease. There are the monoclonal antibodies, which of course our president had when he was hospitalized at the Walter Reed Hospital. This is the product from Regeneron. There's a similar product now from Lilly as well. So that's one course of treatment at one stage of the disease. There's the direct antiviral drug remdesivir, which probably is still a little bit uncertain as to the best place in the course of the disease to use and how effective it really is. And then, of course, there's the other drug, a very long-standing drug, dexamethasone, which is used late in the course of disease if the patient is in a very severe immunological crisis, perhaps on ventilator, and that's a valuable treatment as well. So the course of treatment depends very much on the correct diagnosis of the causative agent. So we need to know that. Then there's the second piece, which is really about public health. So what and really trying to limit the spread of the disease, something which unfortunately in the United States we have not been able to do very effectively. But we start off usually with an index case. This is an individual who presents at the health center. He or she will have symptoms. test will reveal that that individual has got a coronavirus infection. So what do we want to do? We immediately want to reach out and to find those individuals that the infected index case has had contact with. And I just represent 4 individuals here. And time will tell, but what we can suspect is, and I've shown this in green, is some of those individuals will not have been infected from the index case, but some of them, shown in orange, will be individuals who have been exposed, yet may not yet be exhibiting symptoms. So the key piece of public health testing and tracing, establishing contact with individuals and quarantining those individuals, time is very much of the essence. Time to very quickly test and diagnose the index case, and time to very quickly then reach out to the contacts of the index case, identify them and get them into quarantine and to get them tested as well. Because if we don't do that, then maybe the already infected contacts themselves will have other contacts to which at some point in the future they will spread the virus. But if we can intervene quickly enough, then we can block further transmission. So this is, again, the importance of having a test, doing it promptly, getting the results promptly, and being able then to activate the public health process of contacting the individuals who may have been infected by the case in concern. That's certainly been done very successfully in some of the countries around the world, in Korea, in Taiwan, in Singapore. They were able to operate very quickly. They ramped up their testing and were able to limit the spread of the virus in those countries. There were some exceptions, there were some super spreader events, but by and large, they were successful. And in part, I think because they had the experience back in 2002, 2003, of the original SARS outbreak, the one that was notably in Hong Kong and in Toronto in Canada. And with that as their background, they were prepared to respond quickly, whereas in this country, we were unfortunate in not being able to respond quickly. So we test then for diagnosis and treatment. We test to try and exert public health measures to limit the scope of the infection. But there's also a third piece that goes a little bit beyond testing, and that's to do virus sequencing. And this has perhaps not had a lot of exposure in the press, but and it's something that hasn't been done so much here in the United States, unfortunately, but has been done much more frequently in the UK, in places like Iceland and South Korea and New Zealand, who have got their cases well under control. Now, this virus actually has a relatively small genome. It's called an RNA virus. That means its genomic material is in the form of this molecule called RNA, chemically very, very similar to DNA, which is what our genetic material is captured in, but it's an RNA virus. The human genome has made up of 3 billion individual letters. So the background to this is that DNA and RNA each are made up of just 4 building blocks, each one of which has a letter. The human genome is 3 billion letters. This coronavirus, it's only 30,000 letters. So sequencing it is not a problem, can be done very easily. And what I show in that bottom left-hand corner is a little handheld device that you can plug into your computer and can use it to do sequencing. Now, so it's only cost $1,000. So instead of getting that new iPhone, maybe for the holiday gift, you want to get a little handheld sequencer, and then you could have great fun at home. You can sequence the cat, sequence your house plants, whatever you like to do. But it's relatively easy to do now. This is the wonder of some of the technological advances that we've seen. So why do we want to do the testing? Why do we want to do sequencing? Well, two real objectives here. One would be to identify cases that have a common source of infection, and we can do that by looking for the unique signatures of individual isolates of virus. And that will help us to understand how virus spreads, and I'll give you an example in a minute. And then secondly, we want to identify new variants of the virus as they emerge in case they potentially have different properties, which might want us to think again about strategies for treatment or prevention. So let's look first of all at the idea of common source. And I take you back to back to February of this year, which seems such a long time ago, when the company Biogen, it's a biopharmaceutical company based in Cambridge, Massachusetts, they chose to have a substantial, over 100 participant meeting of their senior executives and scientists from all around the world. So 100, 150, something like that, people all converged on Boston at the end of February, just as the epidemic, the pandemic, was getting underway. And what emerged from this very quickly was that of all of those participants, 99 of the conference participants developed an infection with the SARS virus. And there was a paper, and this is where this information comes from. It was just last Friday's issue of Science Journal. So you're getting the very latest information here. Researchers have looked at those cases, and where they were able to get a sample from the individuals who were infected back in February, they sequenced them and found that, in fact, every one of those samples they were able to sequence from the Biogen meeting, everyone was infected with what turned out to be a unique variant, a mutant of the virus called C24116T. We don't need to go into the details of what that means. It actually has a very specific and useful meaning to molecular biologists, but this was a unique mutant found in this case, which says immediately there was probably just one individual from somewhere in the world who came to this Biogen meeting and started this outbreak, a real super spreader event. So as the researchers who wrote this paper in last week's Science looked out beyond the Biogen meeting, they found that the virus spread fairly quickly. It turned up not many weeks later in, for example, a homeless shelter in Boston. So, this is interesting. How does it get from one very high socioeconomic group? I don't think too many of the Biogen executives or scientists have to live in a homeless shelter. So somehow it made that jump across a big social stratum into the homeless group. And it also spread right across the United States. It was particularly prevalent in Florida. And I think we could all understand with the I-95 corridor that runs up and down the East Coast here, how it could get transmitted down to Florida. It's across in many of the states here in the US, and it's now, of course, gone around the world as members of that Biogen community departed from the conference and went back home. So that's one example of how we can trace common-sourced epidemics, outbreaks. The second one is identifying new variants. And again, we go back to about March time for the first example of this, when researchers at Los Alamos Center were doing a lot of sequencing, and they saw the emergence of another, at that time, unique variant, which is called D614G. And this first appeared in Europe at about the time that the pandemic was underway. And looking at isolating and sequencing strains over the following weeks and months, they observed that, in fact, the mutant strain 614G very quickly outgrew and became the predominant strain of infection in Europe. And it was that strain that came across the Atlantic to the United States. It's became the predominant strain in the US, subsequently spread elsewhere in the world, back to Asia, where it became the predominant strain. So the question is, what is the uniqueness about this strain, and is that important to us? Well, what we know is that the mutation for this particular strain is in the virus spike protein. And I think everybody understands that. We've been exposed to so much information. Everybody knows how important that spike protein on the outside of the virus is. It's the mechanism by which the virus is able to attach to a human cell and to be able to get into the cell and start the whole process of infection. So the question is, does this change, this mutation in the spike protein, give us a different virus variant with some new properties? Well, the good news is that in reporting all of this work, the scientists were able to match up a whole sequence database in the UK with a clinical database in the UK, and were able to confirm, at least for the subjects that they were looking at, that the disease caused by this mutant is no worse than what is observed with other strains of the virus. So this doesn't appear to cause a more severe disease, but laboratory experiments suggest that it is more readily able to infect cells. So this may be what the effect of the mutation is in the spike protein. It allows the virus to dock onto cells, to latch onto cells much more readily and get inside and start the infection. Now, just earlier this week, on Tuesday, there was an announcement out of the UK of another new variant, which is increasing rapidly amongst the population, particularly in the southeastern part of the UK. And scientists are looking at this very carefully also because this has two mutations, both of them in the spike protein. One of them is in the very, very tip of that spike, an area we call the receptor binding domain. If you think about, I can give you a nice analogy because you've probably seen this on the news of late. The SpaceX Dragon capsule, there was a wonderful piece of video as the Dragon capsule came closer and closer to the International Space Station and then docked on with a hearty clunk as the two pieces joined together. So the receptor binding domain on the virus is rather like that very finely engineered part of the Dragon space capsule, which allows it to dock in onto the cell and allow the transfer of the components from the virus into the cell. So the new variant is being examined very closely because it does seem to have, again, this propensity to possibly to be able to infect cells more readily. So there's a couple of examples of why testing and of sequencing can be useful to provide useful information. So at that point, Paul, are there any questions?
No, there are no questions right now, so please continue.
Okay, then we'll keep on. We'll move on to the sort of next phase, which is to talk a little bit about the tests. We've got two different approaches. One is to look for the virus itself. That's fairly obvious, I think, why we should want to do that. And again, that breaks down into two opportunities from a technological point of view. We can look for the virus genetic material, which is one sort of test, and then we can look for the virus antigens. Those are the proteins that are produced by the virus in the infected cell. Some of those are incorporated into new virus particles that spread out through the body. Some of them are just within the infected cell, and when that ruptures, those proteins are released into the milieu of the body. So we can detect either of those two as opportunities. The second type of test is really a post hoc test. It's looking for the human immune response, which says the virus was here in the past. So I call that looking for virus footprints. And we now have a number of antibody tests, and we'll be talking about those later on as to what utility they have, which has been relatively small until this point. But I think we'll see much more of that information coming out now that we're talking about vaccines and getting people immune to the disease. So just to put this in context, this is really trying to look at the whole dynamics of various elements and how the tests fit into that. Along the baseline here, what we've got is a line that represents time in weeks. In the middle is time 0, which is defined as when the patient has the first appearance of symptoms. Probably that individual has been exposed to virus anywhere between 1 and 2 weeks previously, and the virus takes time before it begins to get sufficiently of a hold that symptoms begin to be apparent. Approximately 1 week before symptoms become apparent, we can begin to detect the presence of the virus. The vertical axis is a probability across a wide range of people of us being able to detect virus at a given time using any one of a variety of tests. The gray line, the highest peak, is the test that looks for the virus genomic material, the RNA, using a test which we call PCR. And we'll talk about what PCR is in just a minute. That's the most sensitive test, picks up the best. The orange gold line is the results for an antigen test, slightly less sensitive, slightly lower probability of detecting virus, comes up a little bit later dynamics. Then on the right-hand side, you can see the antibody responses. There's two types of antibody that we can observe. There's one called immunoglobulin M or IgM. That's the one that appears first but tails off rather quickly and is replaced by immunoglobulin G, IgG, which persists for a long time. And those two molecules, IgM and IgG, are the focus of the antibody tests, again, which we'll come to a little bit later on. So let's move on. Here's just a little timeline to remind you and to put in context some of the tests that we'll be talking about. Back in January, we had this very unfortunate setback here in the US. So the World Health Organization, when they recognized this was a pandemic, they very quickly recommended to other countries a test which had been developed, I think, in Germany. But here in the US, the CDC said, no, we're going to do the testing using our test. And extremely embarrassingly and very unfortunately, the CDC test initially turned out to be a bust because it was contaminated. These tests are so exquisitely sensitive that the smallest amount of contamination with the wrong sort of agent in the test can really mess it up. So we got off to a bad start, unfortunately, in the US with the CDC testing. It was also proved to be very difficult to get a rapid turnaround, and it was only subsequently that other agencies, including the commercial domestic laboratories like Quest Diagnostics and so on, were able to start doing testing to bolster the total capacity, which the CDC just didn't have. In May, we had the first antigen test. In August, we had what is, we'll come to some of these, the first antigen test that didn't require any piece of equipment whatsoever. I think you can immediately grasp the significance of that. Does not need a centralized laboratory, any sort of laboratory, just to run a test which is based on a small credit card sized piece of paper. And in November, we had just a few weeks ago, the announcement of the first full at-home genomic test. So you can now do it in the privacy of your own home. And we'll come to all of these a little bit later. So viral genome detection and this PCR test or polymerase chain reaction test, this really is the gold standard test. And it's based on actually 3 Nobel Prize winning discoveries. So we start off way back in 1953 in Cambridge, England. On the left is Francis Crick, and on the right is James Watson. And they're the ones who told us about the structure of DNA. They figured out what the structure of DNA is. And just to remind you again, DNA in the human genome, again, as is the case in all creatures in the entire universe here, they all have their genomic material made up of just 4 building blocks. In DNA, those are labeled by the letters A, T, C, and G. And what Watson and Crick were able to show is that in the human genome, it's a double-stranded helix. There's 2 chains, one that runs from bottom to top and one that runs from top to bottom. So we call those anti-parallel, and they're complements of each other. So on one chain where you have the letter A, you will have opposite it on the other chain, the letter T. Where you have a letter G opposite, you will have a letter C. And that allows these 2 chains to bind together and form that stable double helix. Now that turns out to be an extremely elegant solution for when you then want to replicate that DNA. When the cell needs to divide, you need to make a copy of that genetic material. So there's some that goes to each of the 2 daughter cells. And what Watson and Crick and others subsequently were able to very clearly demonstrate is that the DNA replication occurs by you essentially separate the two strands and you make copies of each of the two strands. You make complements of them. So you start off with one double strand, you separate the two strands, and then you can end up with two double strands, which are exactly identical and identical to the original parent. So it was that sort of observation that led to the development of the polymerase chain reaction technology. So if you look at this graphic, and about the middle, you can see the two strands of DNA shown there. And this chap at the bottom here, Carey Mullis, he was the one who eventually worked out the mechanics of doing a polymerase chain reaction test. He was working, I believe, at Cetus Laboratories, which would be on the east side of San Francisco Bay Area. And he was able to develop the basis of the polymerase chain reaction technology, which allowed, in a very simple way, to replicate the DNA from one. If you start off with one single-stranded copy of DNA, if you do this reaction, which is to separate the strands or to melt the DNA so that the strands separate them, you can then join onto those strands little primers of sequence to start the reaction. So you prime it, you then copy it, and you get two strands. So if you go through that sequence, melt, anneal, replicate, melt, anneal, replicate, you do that time after time, you go from one double strand to two double strands to 4 to 8 to 16 and so on. If you do this about 40 times, and if somebody's got a calculator there, they can calculate this for us, you can get to billions of copies of the DNA just by this simple method of replicating each of the strands. So that was the genius of Mullis's discovery and his working of the technology. It's been elaborated a lot more since, but that won him the Nobel Prize. So we're left with one more piece, and I mentioned earlier on that whereas our DNA is in the form of, our genetic material is in the form of double-stranded DNA, for this coronavirus, the SARS virus that causes COVID-19, its genomic material is in the form of RNA, and it's actually in the form of a single strand of RNA. But that's not a problem because back in the 70s, these two guys at the top, on the left is David Baltimore, and on the right is Howard Temin. They were working with what's called RNA tumor viruses, which caused cancers in mice. These viruses turned out to have their genomic material in RNA. And what Baltimore and Temin were able to show was that the viruses contained within them an enzyme which copied the RNA into double-stranded DNA. And this enzyme is called reverse transcriptase. So you put all of this together and we now have reverse transcriptase polymerase chain reaction, or RT-PCR. So now we can think of this whole sequence, starting off with the RNA at the top of the chain, copying it into double-stranded DNA, then replicating that double-stranded DNA through 40 cycles, and you get a very large amount of product at the end, which can be detected by a whole range of different techniques. Fluorescence is one that's commonly used as well. So this is the gold standard assay, and it really is super sensitive. It can pick up one copy of the virus genome in 1 microliter of solution. So it's extremely sensitive. It's extremely accurate as well. So this is the picture of a typical laboratory instrument now that's used for the polymerase chain reaction test, PCR test. This is from Thermo Fisher. You'll see underneath the lid there, which is still open, there's a couple of plates, a blue one and a green one. Each of those plates will hold something like 370 samples. So in one shot, once you've got all those plates loaded up, you put them in the machine, press the start button, you'll be able to test over 700 samples for the presence of the virus genome using this RT-PCR test. And that's done commonly. Now, This test has a number of strengths. As I said, it's highly sensitive, it's very accurate, but it's also, this is not rocket science. It really is established technology. Anywhere, any sort of big institutions here in the Upper Valley, you'll find these types of machines and this type of work being done. Dartmouth-Hitchcock, Dartmouth College, they've got a PCR machine at the Cold Regions Research and Engineering Laboratory. It really is not rocket science. It's just standard molecular biology techniques nowadays. And what it allows to do, as we saw, was you can do parallel testing with one machine. You can do over 700 samples at a time. So if it's got these strengths, why have we had such troubles? Well, it also has weaknesses. The parallel testing is its strength and its weakness. If you want to use that machine efficiently, you can't just test one sample. You've got to load it up with all 750 plus samples before you press the start button. These tend to be in centralized laboratories, so that means you've got to do shipping. You've got to ship your samples from the patient, from the healthcare center to wherever this assay is being run, and that results in delays. And as we'll see a little bit later on, 4 hours doesn't sound long, but nowadays assays are getting down to 15 minutes. So it's becoming a weakness of this big centralized PCR test that it's taking so long to do. It's work, there's no doubt about it, in some countries. Some of this is from China. I show you here, the top picture is people in the city of Wuhan when the virus was first detected. These are all being swabbed and being tested. We saw a lot of videos from China. Some of it were not at all comforting, the people being dragged out of their apartments by health professionals, or call them what you will. Maybe, I don't know what maybe they're police in their protective equipment, dragging people out of their apartments when they thought they were infected to put them into quarantine. The Chinese were able to screen it very effectively and to tamp down the infection. They shut down Wuhan, yes, for a period of time, but they went through the entire population, testing, quarantining, and were able to shut down the infection. There was an outbreak. This is a piece reported in October by the BBC News. There was an outbreak in Qingdao caused by one individual coming in from overseas. They immediately set to test the entire population of the city in only 5 days using this test in order to be able to go through this very effective public health measure of testing, tracing, and contacting. And you can do that in a command economy, a command society such as in China. But I don't think that's the sort of thing that we would accept here in the United States. So what we got here in the US is, well, it's something a bit of you're on your own type of economy. The lack of leadership really from the central administration, from the Trump administration, has meant the states have each got to work separately to figure out how to do this. And what we've seen, and you'll see it even today, you'll open up the New York Times or Washington Post, you'll probably see a photograph somewhere along the line of a lot of people lining up in cars to get tests, and the results take time to come back. It doesn't necessarily happen overnight. It takes maybe 2 days, 3 days, and the result is that with also a lack of resource being applied to do the contact tracing, we still have an epidemic of disease which is completely out of control. Now, it is possible to manage it. And I draw your attention to the results that Dartmouth College have had over the past 2.5 months. When the college announced back in the summer that it was going to allow approximately half of its undergraduates to come back on campus, there was a lot of concern, I think, in Hanover and probably in towns around where students rent accommodation as much as living in college dorms actually in Hanover. there was a lot of concern about the possibility that this would be the beginnings of the spread of infection throughout our community. Dartmouth implemented a very rigorous program of testing, quarantining, and isolating its students. Right from the students who were due to come back to campus were tested before they left their homes, they were tested when they arrived, they were quarantined for a period of time before being released. Students were tested twice a week throughout the term. Faculty and staff tested once a week, so that through the fall term, the college executed over 57,500 tests, and all of those samples were collected and the results were back the next day so that they could take immediate action on any positives. And out of all of those tests, all those students, all the faculty and staff in that term, they say they had 44 positive cases. So it can be done if you put your mind to it, you put the resources to it, it can be effective. Absent what the college was doing, how can we increase our capacity of PCR testing? And I draw attention to a piece by Paul Romer. Paul Romer was the chief economist for the World Bank. He's a Nobel Prize-winning economist. He had an article back in about April time when the whole of the US was essentially in lockdown. About 70% of the population, maybe slightly more, maybe slightly less, but 70% were at home. Only a small proportion of the population were at work because they were considered to be essential workers, whether they were in meat packing plants or transportation or working in supermarkets. There was a small proportion of the population was at work, but the majority of the population was stuck at home. And this clearly was going to be a big drag on the economy. So Paul Romer developed a model which tried to turn that on its head and said, okay, how can we get to a point where 70% of the population is at work and only 30% of the population is in quarantine, being tested, being isolated? And he did some calculations and he came up with the idea that we needed to do 30 million tests a day. 30 million a day in order to get that dynamic of only 30% of the population being in quarantine at any particular time because they were either a defined case or because they were a contact of somebody who was infected. We never got anywhere near 30 million tests a day, but he did offer a potential solution which would reduce the number of tests that need to be done. And this wasn't an original idea. This was actually goes way back to the Second World War, when the US Army was faced with the problem of the outbreak of syphilis amongst the army troops. They wanted to do testing, but they didn't have the testing capacity. And the idea emerged of doing testing of pooled samples. So that's where it began. And nowadays there's a whole area of mathematics around pool testing for everything, not just for diagnosis of disease, but for testing the lights on your Christmas tree or anything else like that. There's a whole series of, there's a whole branch of mathematics related to pool testing. So this is the way pool testing can work. In this example, we posit that we have 27 samples from individuals, only one of which is positive. Now, you could test each one individually, that would be 27 tests. Alternatively, in this example, in method 1, they're pooling 9 individual samples into a single pot. So you now end up with just 3 pots to be tested. You test those 3 pots, you find one of them is positive, the other 2 are negative. So you then go back to the original samples that made up that positive pot, separate them out, test them all individually, and you find your positive case. So now instead of doing 27 tests to find the one positive, you've done just 12 tests to find the positive. Method 2 is just an elaboration of that, an additional step in the middle, but what it ends up with is allowing you to do only 9 tests, just 1/3, 9 tests in order to find that positive case. Now, this only works, admittedly, when the number of positives are relatively small. If you've got a large proportion of your samples being positive, then it does not become a particularly useful method. But this is nonetheless can be done and was done in Wuhan when they had a further outbreak in May, and they processed 2.3 million samples very quickly and were able to identify 56 new cases in the city. And eventually, in July, the FDA decided to grant company Quest Diagnostics approval to do pooled sampling. There's rather late in the game, but that was eventually recognized as being an option. So that's one way to increase our capacity. Scientists have also been looking at new technologies. Rather than using the PCR test, are there easier ways to be able to detect the presence of the virus genome? And this gave rise to something that is called LAMP, or loop mediated isothermal amplification. We don't have in this system to heat the sample up to separate the strands, let it cool down, replicate them, heat it up, let it cool down. This is all done at a single temperature. And we don't need to get into the details of how all this works, but what it means is that it does provide a test which is relatively easy to perform, is relatively cheap to perform, does not need a lot of sophisticated machinery, so would be ideal for use in developing countries. And this is now actually becoming a test which is being applied, as we'll see, the technology in a number of tests. Then there's the method of detecting the product that's produced by amplification. There's a whole range of ways in which this is being done. New things are being added all of the time. The gene editing technology called CRISPR has been brought into play here, got approval from the FDA, just as an example, again, of lots of different new technologies that are being developed. They're not solving necessarily the problem that we're faced with today, but in the future, you may well see these technologies being applied more generally in clinical medicine. And so one of these, the LAMP testing, has given rise to what is now at the point of care testing and something called the toaster. In March of this year, the FDA granted approval for Abbott to do a single sample assay. And it uses a machine like this, which is in fact widely available as a point of care testing machine. If your grandkids get taken to the pediatrician because they got a slight fever, you want to know if it's, if it's strep throat or it's flu, it could be this type of machine that the doc will take a sample from the child, put it into the machine, and 20 minutes later, you got your answer, whether or not this kid should be getting antibiotics for its strep throat, or should be getting perhaps something like Tamiflu for influenza. And it uses this isothermal technology to shorten the time from days with the PCR where it's being sent to a central laboratory to just 20, 30 minutes in the doctor's office. And a number of other companies like Becton Dickinson and so on have got these types of machines. This is the type of thing that would be in the White House. Anybody who's coming into the White House to meet with the president, we're told, has to be tested first for evidence of if they make sure they're not infected with the COVID virus. This is the type of machine that would be used to test for them. It has some downsides. It's not as sensitive as the PCR test, but that's outweighed by the speed with which you can get a result. And so there's a good trade-off to be made there. Then, as I mentioned, I gave you a little feel for, we now have our first in-home, fully in-home genomic testing. And it works simply by, you're given all of the kit that you need in this, you take a nasal swab, you put it into a little vial which has got some fluid to extract the genome from genomic material. You put that into the test system, which is a little holder that's driven by batteries. It's a 30-minute test using this isothermal amplification, and you get a simple readout, not quantitative, but just a simple readout. You're either positive or you're negative. So this is, again, a great step forward. Does it solve the problem? Not entirely, in my view. You must have a prescription first from your physician. So this is not something you're going to do every morning when you wake up and before you decide whether or not you're going to go out to your office or to some other event. You need a prescription. It's also a $50 kit. And, you know, that's a lot to do sort of routine testing with. But it's good, there's no doubt about it. The data show that it'll give you about 94% agreement with the gold standard PCR test. And its weakness is when there are very low levels of virus. It tends to have more false negative results then. It tends to miss the low level cases. The number of false positives, that is, you know, it says you are infected when you're not. Those are relatively low also. That's a bit of a bear, of course, to get the wrong answer and to think you are infected when you're not. But that, fortunately, is relatively rare. So this is a good step forward, but doesn't solve our problems entirely. Now, I just wanted to put this one up. This, again, is from this month in an article in Nature. Some additional new technology, fast thermocycling with plasmonic heating through magnetic plasmonic nanoparticles. I mean, this is just technology going to the extreme now. Again, it might be useful in the future. It may become the standard for a lot of diagnostic testing. But right now, we don't need any more technology. We need more implementation. So just to wrap up this section, this is, you know, a typical sign, maximum tests reached today, come back tomorrow. This is really no way to run a system. And there was a paper from a group led by Larimore et al. They did some modeling early on and said that what's really important here is speed. People have got to get able to access the tests, access them quickly, access them frequently, get the answer back promptly. And all of that should be the priority. To have an exquisitely sensitive test should really be secondary. The PCR test does not, as it's currently executed, meet the goals that Larimore et al. Put out. It meets the secondary goal of being wonderfully sensitive, but it does not give us the turnover. So what we need is not this Cadillac test, of which there's just a few examples out there, but we really need, you know, lots of the basic Fords or Chevys that can very quickly give us the answers that we're looking for. And so we're going to go on to that, but just to pause, are there questions, Paul?
No questions yet. Nope. Ready to continue.
Okay. We'll move into the next section. So this is a lead into the idea about antigen testing. I just wanted to show you this graphic, which I got from John Sanders, who I think presented to you recently. And you may have seen it before in John's presentation. Again, along the bottom is line. The bottom line is time, probably about week and a half, two-week intervals. And it shows how the virus load changes in the individual over time. It begins to rise in the before evidence of infection. It actually begins to rise before you can begin to pick it up with the PCR test. Then it peaks and then tails off over a longer period of time. If you're using a highly sensitive technique like PCR, which is shown as that bottom dashed line, you have a very wide window in which you can pick up virus presence. So testing infrequently becomes less of a barrier to actually identifying a case because you've got a big window of time in which to catch them. If you've got a test with a lower analytical sensitivity, which is the dashed line, the higher of the two dashed lines, the window in which you can detect virus is much narrower. The window of time is much narrower because you can only detect higher amounts of total virus present in the patient sample. So in order to be effective, you just need to do more testing more frequently in order to be able to catch that window of time. So this is where the antigen assays begin to come in. They're cheap, they're fast, they're certainly less sensitive, but they can be done more quickly and more readily and use, as you'll see, they start off by using instrumentation, but in fact, the latest developments, we don't even need any sort of instrument. So the first one was approved by FDA in May. It's based on some fairly standard strip test technology. It was a 15-minute test, but it still needed a machine to read it. What might have been the game changer was this release by FDA in August that they were authorizing a diagnostic test which could be read directly from the test strip. And what this is from Abbott, and it's a credit card sort of size test, piece of test board on which you place your sample, allow it to develop, and then to read out the result. So the health, as defined by the Food and Drug Administration approval, still requires a healthcare provider to perform this test, to swab the patient, apply the sample to the card with the testing reagent, wait for 15 minutes and read the card. 1 line equals negative, 2 lines is positive. Not rocket science. I think we could all do that. But at the moment, this is still, the FDA requirement is that it's done in a healthcare setting. Can't do this at home. The government thinks it's good. They said that they were going to purchase 150 million of these test kits for use in schools, to test kids, and for use in nursing homes. And I guess in both of those two settings, you can have a healthcare person there to execute the tests on the patients or on the subjects. And if you go to the Abbott website, this is the lower part is the picture you'll see on the front of their website for taking these COVID-19 tests. This is their vision for the future. What it's showing here is, I don't know, this could be the entrance to a college building or to an office. We've got some young techie type people coming in. One of them's got his bike with him. You know, they're still wearing their masks. The guy with the bicycle has got his phone in his hand, and what that phone has on it is an app. If he takes this test using the test card, you can then transfer the data onto your phone, and your phone can then act as, just like on an airplane, where you see people who are boarding the plane, they have their boarding pass on their phone, so you could have your virus antigen test results on your phone that say, this person was tested on this date and is negative, and whoever manages this building can set their own parameters for who they let in. But you just show your phone, bing, you can come in, come to work, go to lectures, whatever the circumstance might be. I think this represents Abbott's vision for the future. We're not there yet, but some of these elements are coming into play. It's a $5 test. It only costs 15 minutes. It's beginning to have all the elements that we need of affordability, of speed and simplicity. And obviously, this will be readily accessible. You can download the app from the various places where you get your apps. And I'm sure that Abbott is working with the FDA so that this test can be applied in the home setting in some time in the not too distant future. These are moving towards more and more towards the sort of assay setups that are used for, say, pregnancy testing. We've got a lateral flow antigen assay, which has come from Innova. This is now being used by individuals. The UK government is running a test on this in a large city. I believe it's the city of Liverpool. They're giving out these tests to all the people, for the people to use for themselves at home to see whether or not they have a positive or a negative at the start of their day, and they behave appropriately. The reason why it's controversial is based on these data. The company provided 3 sets of results for using the test, using 3 different test conditions, and those are shown in the 3 lines. The first one was using trained laboratory staff. So those will be individuals probably who actually developed the test in the first place, so they know exactly how it works, they know exactly how to apply it. And that, using a whole series of samples, they showed that the sensitivity overall of this test was about 77%. If you looked at samples which had very high levels of virus in by the PCR test, then it was about 95% sensitive relative to PCR. When you looked at trained healthcare staff, the level of sensitivity, the ability to detect samples, dropped slightly from 77% overall to 73%. But when you then let this loose on the public, which essentially are self-trained, you probably get a little sheet of paper which tells you how to swab, how to extract, how to put it onto the test card and so on. The sensitivity dropped to 58%. So this is getting, I think, into the realms where it becomes less and less and less useful. And that's actually led to the FDA being very cautious, and it's approved this type of assay from CareStart, but it's, again, still only for use in a clinical laboratory. Must be applied by a trained healthcare specialist. And this is where we might have left it, except that over the last couple of days, we've now had announcement of a further approval of a lateral flow assay, reported just, what, 3 days ago. The FDA authorized the first fully at-home diagnostic test from a company called Ellume, E-L-L-U-M-E. They're based in Australia, but I think they also have laboratories in California. And here's the similar set of results for what they found to be the case. It's in the simulated home setting, so that would be done in a laboratory, but with something that simulated the home setting. They had the sensitivity of 95%. So relative to PCR, they picked out 95 of 100 positive tests. If it was symptomatic disease, the sensitivity was a little bit higher. If it was asymptomatic disease, the sensitivity level dropped to about only 91 out of 100 tests relative to PCR, but this is still pretty darn good. And so this has been approved now by FDA for at-home diagnostic use. The challenge is it's going to take time to roll this out. We need millions of these tests, and that probably won't be happening until about the summer of next year, according to the company's press release. But I think you can see what's coming down the road at us, and this could be still useful, even in the context of a vaccination program. We all very hopeful for the vaccination program, but not everybody is going to get vaccinated. Not everybody may want to get vaccinated, and so there may be indeed a place for this type of home testing. I'm sure Abbott, seeing this, is going to be striving very hard to get their test approved for use at home. A $5 test at home that you can then put on your iPhone or your Android phone and show that as you go into a building, onto an airplane, wherever it might be, is just going to be the real answer to this. So that's antigen assays. I have, I think, about 2 more slides. So we'll just quickly go through antibody testing. It's not something that has got a lot of attention up until now, but it's going to become more and more to the fore as we go into this program of vaccination. So what do they measure? Now, it's very important here to understand the differences between the various tests. What's loaded up, and the picture at the bottom shows the sort of test strip that's used, a little plastic gizmo. You put your sample in, you let it flow up a little paper strip, and then it causes lines to appear in the window. The paper strips are loaded up with virus antigens. Now, commonly, a number of these tests use either the N antigen, that's one that's part of the virus capsid, the virus particle itself, or P antigen. And the reason why these have been chosen is they're relatively easy to produce using standard biotechnology fermentation processes. You can produce these proteins in quantities and load them up onto millions of these test strips. It's easily done.
Okay.
But the disadvantage is that this test only then detects antibodies after a moderate or severe infection. The second type of antigen, which is much more useful, is the virus spike antigen or S antigen. But this is tough to produce in biotechnology processes. So it's been less of a focus for these test kits up until now. But this is far better because it'll detect antibodies that come up after an asymptomatic or a mild infection. And of course, it's going to be useful for testing people after they've been vaccinated to understand their level of infection, their level of antibody response. So this is the sort of uses. Of course, it tells you if you've had an infection, maybe even how recently your infection was, and it can be used to screen immune plasma. We've seen a lot about using immune plasma from recovered cases in order to treat those in hospital. So it has some application there as well. But what do we want to do? What do we measure? Well, we really want to understand how many people in the population have been infected. And quite frankly, at the moment, that answer is not really known. It's clear that there are a number of people who've been infected and have never had symptoms at all. The most rigorous analysis suggests that 17% of the population have had asymptomatic infection, but that's based on individuals who presented for a test, a COVID PCR test, were found to be positive, but being followed for weeks afterwards, they never showed infections. What it doesn't capture is those individuals who were infected, never went to the doctor's office, never were tested, had an asymptomatic infection. We'll be able to capture those now using antibody assays. We also want to really know to what extent do these asymptomatic people contribute to the spread of virus. The information suggests that it's rather limited because the window in which they have a positive for virus is much less when you're asymptomatic than if you've got symptomatic and even severe disease. And also in children, the evidence seems to suggest that children tend to have asymptomatic disease and probably are not big spreaders of the virus, which would be really, really good information to have and good news. The other thing we want to know, of course, is that people with antibodies, do they get reinfected? Right now, that seems not to be the case. Reinfections seem to be very rare. The outcomes are variable. We can't really say too much about what is a common pattern right now, but that's going to be good information to have. And then secondly, in the context now of vaccination, what we're going to want to know is how long does that immunity, which is given to us by the vaccine, how long does it last? So we can measure antibodies using these types of tests. And people have been doing that already for a while for individuals who were infected. And the news has been pretty good, at least up till now, for those studies that have been performed. And we've only had a limited amount of time since the virus even came on the scene, but people are saying that antibodies seem to persist for 3 months, 4 months, 6 months, which is good. Your immunity levels are probably still there. But we'll need to answer this question in the context of once you've been immunized, how persistent is your immune response? Do you need to have booster shots of the vaccine, maybe once a year, once every other year? We just don't know at this stage. It may be that 2 shots, 2 and you're done. You may be good. We just don't know right now. So that's how antibodies, I think you're going to see more about antibody assays in the press as time goes on, as more and more people are immunized, and we're asking these sort of down the road questions. So with that, I think that's all that I have for you for today. I thank you very much for participating and listening. And I would say that, again, if you find this and other talks like this from my OSHA colleagues enjoyable, please do look at the OSHA website and look at our catalog of courses. I may be doing something again in the spring term, haven't decided that yet, but we'd certainly love to have you join in. And of course, you can join right now on Zoom. And who knows, in the future, maybe also we'll be back in in-class courses come the fall when we've all been immunized. So I think I've been asked to show this slide from Sue. If you have any other questions, what to do, suggestions for topics.
There are a couple of questions for you.
Okay, so let's have those. Let me see if I can stop sharing. Okay, far away.
Okay, first question from Ken Dolkart. Do any of the antigen tests measure spike protein? And if so, is there a crossover false positive after receiving the mRNA vaccine?
So it's not clear to me how many of these tests, these new tests, use the spike protein. The information is not widely shared, and it's if you like something of a trade secret. Now, the way the new vaccines work is that they cause your cells to produce spike protein. So if you were using an antigen assay and you had just been immunized, you could probably pick up that spike protein circulating in your blood or in the samples. But it's not the same as an infection, because the virus, when it infects you, it will infect not just your nasal passages, not just your lungs. It can infect cells that line the blood vessels, the gastrointestinal tract. So in a normal infection, you've got virus spread throughout the body, the potential for that to happen. With the vaccine, it's essentially restricted to that area where the jab was actually given. The way these vaccines are made is it's a piece of RNA, the virus genome, just a small stretch which codes for spike, that's captured in a little fatty globule. Those globules are injected into you, they enter your cells, they start to produce the spike protein, but they don't spread to other cells. It's only in those first few cells that are exposed to the injected material that will produce the spike protein. To the extent that it's expressed on the surface of the cell and maybe is lost into the bloodstream, but I don't know, I don't think probably you're going to see very much in terms of picking that up in an antigen assay.
Okay. And we have a second, keeping it all in the family, with Vivian Dolkart. Can home tests shorten the duration of routine quarantine prior to visiting family?
So this is the ever-changing, and it's okay, but the recommendations from CDC, as we learn more and more and more, what you're seeing now is quarantine recommendations that say, if you've quarantined for 7 days, if immediately prior to that quarantine you had been exposed to somebody, you would expect by 7 days that you would have detectable virus by the most sensitive methods. So what the recommendation now is quarantine for 7 days and test. And if you have a negative test, that says that it's pretty high probability that you are not infected and you're not going to show anything in the next couple of days, next week or so. So 7 days plus a negative test is now what's becoming a commonly recommended quarantine regime now. The CDC, I think, has suggested that without a test, it might be just, instead of 14 days, it might be just 10 days is enough. There's probably going to be outliers, but I think, I mean, my family have been talking about this, what should we get together for Christmas or not? and talking about 7 days, then get a test as being a possibility. It's still up in the air, but yeah, I think that's the direction we're going in now.
And I think the final question is relates to the in-home testing trend, if you will. It certainly has advantages with low cost. There's a rapid turnaround. There's no cost for MD prescriptions. How do you see these connecting with public health for follow-up measures?
Well, for example, the Ellume test, which also has an app on the phone, that app is programmed to send the test results to public health authorities. I'm not sure how it works exactly, whether or not people would feel they want to accept that their data, the results of their test, is being shared with public health. But that's something that's apparently built into the app for the Ellume test. That was the last one that I showed you. Yeah, that's a direction in which we're going.
Well, that's it for the questions. I'll turn it back, I guess, to Sue.
Okay, thank you for really fascinating, very interesting talk, Iain. And as we said before, and I'll just mention it again, we welcome your feedback, your evaluation, your suggestions, because we have this program now going out into the end of March. So we welcome any kind of feedback, and that is at eastmancares@eastmannh.org. So please feel free to do that. And now go enjoy the beautiful winter snow, I guess. Thanks.
If I may just say thank you for giving me the opportunity to present to this. I've really enjoyed doing it, and wish you all well.
Thank you all. Thanks for joining us.
Thank you.